Hello everyone. After class today, I looked over the activities that we covered, and there were some areas that I wanted to make sure that the main take-home points were clear. In some areas, I feel like I might have rushed a little bit. Now, usually if this happens, I just make sure it's clear in the next class, but our next class is our test, so I thought it would be a good idea just to make a quick podcast over today's activities, or at least the main take-home points from today's activities. I want to begin with a very quick interpretation of how Robertsonian translocations can lead to an inheritable form of Down syndrome. Usually Down syndrome is caused by a non-disjunction event that occurs with chromosome 21. But in this case, I want to talk about an example where this translocation copy of chromosome 21 that's translocated to chromosome 14 due to a Robertsonian translocation. This cell is from a person who is healthy. They have 2 copies of chromosome 21 and 2 copies of chromosome 14. It just so happens to be that this copy here contains all the information on a single chromosome, the translocated copy. Now, these chromosomes during meiosis can line up in different ways. Don't worry so much how they line up to create the problem that we're going to discuss in just a moment. Just recognize that they can line up and pair up differently in meiosis. As a result, we can get one of several different possible gametes that come out. The first 2, these will result in an offspring that is healthy. Okay. So this middle row here, let me back up and just remind you that these are examples of gametes that are produced through meiosis. Down here are the progeny that are formed when these gametes are fertilized with opposing gametes that have the correct chromosomal content. So over here, they can line up during meiosis where you get 1 copy of each, just like you would normally expect. When it's fertilized with a gamete, another gamete, we will get a progeny that is healthy. 2 copies of each, 14 and 21. Here, this translocated copy here, you have 1 copy of 14, 1 copy of 21. When it's fertilized with a gamete of correct chromosomal content, you will also get a progeny that is healthy because it contains 2 copies of 14 and 2 copies of 21. It just so happens that this one set here are on the same chromosome because of the translocation, the inherited translocation. Now, over here, you could produce gametes due to the way the chromosomes line up during meiosis, where this gamete contains 2 copies of chromosome 14, 1 copy of 21, shown here in red. When it is fertilized with another gamete that has the correct chromosomal content, we will have 3 copies of 14 and 2 copies of 21. This is a trisomy 14, and that will lead to a lethal condition. This will not result in a viable progeny. You could also have a situation that counters this one, where you only have 1 copy of chromosome 21 and no copies of 14. When this is fertilized with another gamete with the correct chromosomal content, you'll end up with a monosomy 14, a progeny that only has 1 copy of chromosome 14. This is also lethal. Now over here, this is the part I really want to talk about because this explains how Down syndrome can be inherited from a parent who is healthy. This first gamete that could form, based upon how they line up during meiosis, has 1 copy of chromosome 14 but no copies of chromosome 21. When this one is fertilized with a gamete with the correct chromosomal content, you will have a progeny with 2 copies of 14, which is good, but only 1 copy of 21. This is a monosomy 21, and that is also lethal. Next, the last possibility we could have is 1 copy of chromosome 14 in the gamete, which is what you want, but you have 2 copies of 21, one that is on this translocated copy of the chromosome here. When it's fertilized with a gamete of the correct chromosomal content, you will end up with 2 chromosomes 14, one of them is attached to this Robertsonian translocation, and 3 copies of chromosome 21, one on the Robertsonian translocated chromosome and 2 independent copies of 21. This will result in an inheritable form of Down syndrome. Okay, now let's talk about the activities we did today in class, at least a couple of them. In this particular activity, we talked about liver cancer and how the virus hepatitis B can infect the liver and how that increases your chance of getting liver cancer. The main thing I want you to focus on is the role that P21 has in liver cancer. How when P21 doesn't work properly, how that can lead to cancer. So this was the activity. I'll leave this up here real quick, but then I'm going to move to a whiteboard. But what we're going to do is briefly go over the main take-home points from this activity. So I'm going to move to the whiteboard now. Okay, before I address this activity fully, I want to talk about what happens normally with all the proteins that are involved. And then we'll talk about what happens when P21 doesn't work properly. So let's begin with a piece of DNA here. And let's mutate this DNA. Now remember, the protein that comes along here that detects this mutation is called P53. Remember, P53 will detect the mutations and decide whether or not it's worth fixing. In this case, we're going to decide that P53 will fix the mutation. And what P53 will do is it will activate P21. So now P21 is activated by P53. So what P21 will do is it will inhibit a specific CDK cyclin. CDK cyclins, remember, are important in turning on the cell cycle. But P21 inhibits it. Since cyclin CDK is not active, it will not be able to do this next step. And what it would normally do if it was activated is it would phosphorylate a protein called RB for retinoblastoma. But it can't do that now, so it's not phosphorylated. And as long as RB is not phosphorylated, it will remain bound to this other protein, E2F. And as long as E2F remains bound to RB, it can't do what it would like to do, and that is to go into the nucleus. So here's our nucleus. So remember, what E2F would like to do is would like to come into this nucleus and activate S-phase genes, or genes to release it from that G1S checkpoint. So it would usually come in here and bind to this promoter and activate the transcription of these specific genes to enter the S phase. But it can't do that. That's what it should do if everything's working well. Then P53 will facilitate the correction and the fixing of these mutations, and then everything is reversed and the cell cycle can continue. However, if P21 is mutated, so it's not functional, what happens is it's not working. So if it's not working, cyclin CDK now becomes active. Since it's active now, it will add a phosphate group to retinoblastoma. If retinoblastoma is phosphorylated, E2F can now be released, and if E2F is released, it can now go into the nucleus and bind to this promoter. When it binds to this promoter here, it will activate these genes and turn these genes on that are necessary to get past that G1S checkpoint. And so now the cell cycle will continue even though this mutation here has not been fixed. So now this mutation will continue with every cell division. And now, since it's not stopping at all, more mutations can accumulate. And ultimately what could happen is that cancer could form. So remember, any gene like P21 here, whose job is to stop the cell cycle. So P21's job is to stop the cell cycle, so it is a tumor suppressor gene. E2F, on the other hand, its job is to drive the cell cycle forward. So it is a proto-oncogene. Now what I'd like you to do on your own is to think about how other genes, when mutated in this pathway, from P53 to P21 to cyclin to retinoblastoma to E2F, how they can be mutated in various ways, identify other tumor suppressor genes or proto-oncogenes, and ask how would that affect the movement of the cell cycle. We also talked about something called gene therapy. But gene therapy essentially replaces a mutated gene with a non-mutated gene. Now there are a lot of technical problems with gene therapy, but let's remove those technical barriers and just say, if it's possible, could that fix this problem here? And the answer is yes. Gene therapy could potentially help replace a tumor suppressor gene mutation. Because remember, tumor suppressor genes, when they're mutated, they're no longer there. So we've removed P21. It can't do its job now. But if through gene therapy we can insert a new P21 gene it could now replace the one that's missing, and in theory, it would drive this forward. Again, there are some technical problems, but just on the principle alone, it should work. Now, however, if E2F has become an oncogene, it's overexpressed, it's always going to be driving the cell cycle because there's too much of it for retinoblastoma to control it. If you try gene therapy with E2F by giving it a new copy of E2F, it's just going to exacerbate the problem, make it worse. And so gene therapy, in theory, would not work to correct an oncogene mutation. Later on, I talk about a suppressor of this. And the answer to that question was that it involved epigenetics and the gene DNMT3 that would have been the other gene mutated. Don't worry too much about that. But what I would want you to know, and review your notes from a previous podcast, is the difference between an interspecific and an intraspecific suppressor. Those would be important things to know. Now, I want to talk about the very last activity, and we actually didn't talk about this in class, so this is bonus knowledge. Lucky you. And I want to talk about it now because it involves a lot of things we have written on this whiteboard. I'm going to erase a few things, though. Okay, so let's go back to our previous state where we have a gene mutation. P53 detects the mutation. It activates P21. P21 works on CDK to make it inactive. So CDK cyclin can't work now, so it cannot phosphorylate RB to release E2F. And this is what we want to happen. In activity 4, again, we didn't get to this in class, but let's talk about it real quick. It involves HPV, the human papillomavirus. HPV is one of these viruses that can lead to several kinds of cancer, including cancer of the cervix, vagina, penis, pharynx, and anus. But how does HPV cause these cancers? HPV causes these cancers by making two oncogenes. And these oncogenes are called E6 and E7. And again, remember, these are both oncogenes. Oncogenes, because when these are expressed in a human cell, they will drive the cell cycle forward. So by definition, they are oncogenes, even though they are not human genes. So what do they do? Let's start with E6. What E6 will do is it will inactivate P53. Now we can't activate P21, so cyclin CDK remains active. If cyclin CDK is active, we'll add this phosphate group here, release E2F, and E2F will come into the nucleus. All right, now let's talk about E7. So now P53 is still working, but E7 will come along here and bind RB. And as it binds RB, that releases E2F. E7 blocks the binding site essentially of where E2F would normally bind. So now E2F can enter into the nucleus, bind to the promoter, and turn on these genes. So what this virus does, it doesn't just do that, it does both of them, right? So not only is RB inactivated, but P53 is also inactivated. It does both. So it effectively, by two different mechanisms, is able to drive the cell cycle even in the presence of mutations. Okay, the last activity I want to talk about is activity 3. And in activity 3, we talked about a specific cancer called Burkitt lymphoma. It's a cancer linked to our immune system. The cells that are designed to help protect us. It's linked to a particular gene called c-Myc. c-Myc is a proto-oncogene. It turns out to be one of the genes that's turned on by E2F. Once Myc is activated, it would help drive the cell cycle forward as well. The way a proto-oncogene becomes an oncogene, that is, the way it becomes overexpressed, can be divided up into 3 different ways, which is mentioned on a previous podcast and we mentioned in class, but let's just go ahead and list them real quick. We can have a point mutation, a missense mutation to be more specific, that changes the shape of that protein that's being coded for in such a way that it is overactive. We could also have that gene c-Myc or another proto-oncogene, we could have it be amplified so that you have multiple copies of it. The last one is by translocation, and I want to talk a little bit about this method. Now remember, these are occurring in our immune system cells. Now more specifically, B cells. And what I want you to know about B cells is that B cells, one of their jobs in the immune system is that they make antibodies to fight off infections. So let's talk about what's happening inside of this B cell. And let's talk specifically about two different chromosomes, chromosome 8 and chromosome 14. Chromosome 8 contains our friend c-Myc. Chromosome 14 contains an antibody gene, more specifically IgH. So what do these antibody genes do? They're important in protecting the cells or the body against various kinds of infections, but they are also important in fighting cancers of the body to stop cancers from growing. So under a normal circumstance, if a cancer cell is detected, antibody genes will be turned on to make more IgHs to protect the cell against these cancers. Okay, so now how can this lead to cancer? It can lead to cancer due to a translocation. And this translocation occurs between the c-Myc portion of chromosome 8 and the antibody portion gene from chromosome 14. And so we end up with two translocated copies of chromosome 8 and 14. That might look something like this. And so what's happened now is we placed c-Myc over here, and we've placed the IgH gene over here. That by itself doesn't seem like a problem, but the problem comes that c-Myc now is under the promoter of IgH, and likewise, the translocated copy of IgH is under the control of the promoter for c-Myc. Now, when a cancer cell is detected, or another infection, this B cell now wants to produce more antibodies. So by activating this promoter, it is trying to activate IgH, but since c-Myc is there now, it's activating the gene c-Myc. So we get increased expression of c-Myc. That's going to cause this B cell to divide out of control. What complicates the matter even more is that as cancers form in these cells, because it's overexpressed, the cells are dividing really fast, so they're going to accumulate mutations, and that's going to lead to cancers in these B cells. The cell is going to recognize it's growing too fast. So it's going to inactivate this promoter, thinking it's inactivating c-Myc, but in reality, it's inactivating IgH. I'm going to restate that again. This translocation event does two things. As mutations are accumulating in these cells, instead of turning on the IgH gene to kill the cells, for the immune system to kill the cells, it's turning on additional genes that are going to cause it to be overexpressed even more. Likewise, instead of turning off c-Myc like the cell thinks it's doing, it's actually turning off the one gene that could help protect it. By doing this, we overexpress c-Myc, and thus the cell cycle is in overdrive, and we downregulate the immune system, the IgH. Because of these two events, these B cells will grow out of control and not make the antibodies necessary to protect it. That's all I wanted to review from today's activities. I hope that helps. If you have any questions over this material or any other material for the exam, please make sure you send me an email or stop by. Good luck, and I'll see you next week. Bye.